gives me great pleasure to introduce our next speaker, Robert Nussbaum, who's the current Chief Medical Officer at Invitae Corporation. Dr. Nussbaum was most recently the Chief of the Division of Genomic Medicine at the University of California, San Francisco, and has played a leadership role in cancer genetics and prevention programs and in the program in cardiovascular genetics. As a board-certified internist and medical geneticist, Dr. Nussbaum has dedicated his career to improving the care of adults and children with hereditary disorders. And today he'll be giving the keynote address on genetic sequencing or gene sequencing and newborn screening. Thanks for coming, Dr. Nussbaum.
Thank you. Thanks very much for having me. Is this working okay? Push the button. Tell me to push the button.
All right. Is it working now? Can you hear me? No.
Try now.
Okay, it's now green, and it sounds right. Thank you. Well, thank you very much for the invitation to talk. I'm going to be primarily wearing my hat as a former PI, but now as a collaborator on a project that was one of the projects that came out of the National Institute of Child Health and Development and the Genome Institute, looking at issues around the use of DNA sequencing in the newborn period. And I've got the PI currently is Dr. Jennifer Puck, and I'm going to show you at the end a very large group of people at Berkeley, UCSF, and elsewhere, University of Washington, who are participating on this project. Okay, these are my disclosures. Obviously, I'm an employee and stockholder in Invitae, and I consult for Pfizer Rare Disease Unit and Genome Medical. So, first I want to talk a little bit about the fact that this project relies very heavily on the excellent California Department of Public Health effort in newborn screening. It's really, it's something like 1/8 of all the births in the country occur in the state of California. And we've had wonderful collaboration, particularly with Dr. Robert Currier, who just recently stepped down as being director there. And he's a close friend and an excellent collaborator. So this is the screening performance in California over a 5-year period that became the subject of our grant. And that was how many specimens were looked at. It was 2.2 million, of which about one-tenth of that, in addition, actually came from NICUs. So these were children who were premature. And why that's important, some of you know and will become obvious later. The number that were positive by MSMS was overall 0.17%, but you can see that although there are 10 times fewer NICU patients, 10 times larger percentage were positive from the NICU because of issues of what's going on with these children in the NICU. The number of diagnosed metabolic disorders, you can see, and the positive predictive value of the screen turned out to be about 17% for the regular, but only 2.5% in the NICU. And what's wonderful about the California program is when there's a positive, these children are brought into metabolic centers. The diagnosis is then confirmed through a variety of methods. There's dietary and biochemical methods, et cetera. But then those children are all followed for the first 5 years. And those metabolic centers are asked to, very, very firmly asked to collect clinical data and get it back to the California Department of Public Health so they can follow up. So we have very good information on what the long-term follow-up was, at least for the first 5 years, for these children, and whether the diagnosis that was suggested by the screening test actually turned out to be confirmed or was ruled out. There are some major challenges facing MSMS newborn screening. The false positives, as you can see. The rarity of the disorders means that there are false positives despite excellent specificity. And it's even worse when you're screening low birth weight or premature infants, and that's why the positive predictive value drops so far. There's also reduced specificity for some results that are actually true positives. And that's because MSMS results provides a differential diagnosis, even in a patient who's affected. And so that and as I'll show you in a minute, sometimes you cannot make a definitive diagnosis, even after, with follow-up of these patients, if you're not just relying on the MSMS, but also there may be difficulties figuring out what's going on even when the patients have been followed for a couple of years. And I'll show you a couple of cases of patients where by doing genetic sequencing, we actually showed that the diagnosis that was confirmed at the metabolic centers was actually incorrect. So that's the false positive problem, is you've got this positive predictive value that ranges from 2 to 40% for different metabolic disorders. MCAD, for example, here's just one example, 18 true positives, 132 positive screens, 92 were false positives, many of which came from babies whose birth weight was under 1,500 grams. There are also 4 heterozygotes that scored positive on the MSMS screen. 18 expired before we were able to do the 5-year follow-up. This was a paper published by Hall et al. In 2014. You can improve the predictive value, the positive predictive value, by using ratios rather than just a single analyte. And of course, there's a lot of work being done by the MBSTRN and other sorts of groups, the group out of Mayo that's looking at this. But there's still a problem with reduced positive predictive value for many of these metabolic disorders. So why do false positives matter? Of course, it's very obvious. First of all, there's the expense. The professional time in obtaining repeat specimens for retesting and follow-up and additional testing. And then furthermore, it prolongs the time to diagnosis, and that creates a lot of parental anxiety. I just recently was a grandfather in the last year. 2 grandchildren were born, and I can just put myself in the chair of my son and my daughter, who go through pregnancy, have a baby, everything seems fine, and then you get this phone call. There's something wrong on the newborn screen. And we don't know if it's real or not, it needs to be tested. So you've got this whole prolonged period of anxiety that occurs. And that can go on, as I'll show you, not weeks, but actually sometimes months. So there's a lot of expense and hassle to the family while therapy is instituted prior to being sure there is a diagnosis. So here's an example of the reduced specificity problem. You get the same abnormal compounds that are increased in different inherited disorders. For example, the C3-acylcarnitine occurs in at least 4 different situations, 3 of which are, or more than 3 of which are inborn errors, but also dietary deficiency of B12. There's also locus heterogeneity, 34 positive screens for glutaric acidemia type 1. However, it turns out that of those 34, only half were really glutaric acidemia type 1, and the others were other inborn errors of metabolism that had similar kinds of tandem mass spec pictures. And the time to diagnosis is important for these false positives. PKU is one of the easier ones to work out, so the mean and median time to figuring out whether something was a false positive or not for PKU in 56 PKU patients was a matter of 10 to 11 days to almost 2 weeks. But MCAD, in 131 of them, you can see the median was 36, but the mean was 63.3. And the mean is so much higher than the median because there's a tail that's stretching quite far out of patients where it took a long time to really figure out whether this was a true positive or not. So, what are the possible roles for DNA sequencing in newborn screening? You could think that you could do primary population screening, a sequence as a standalone test. Now, I put that up there pretty much as a straw man. The idea that DNA sequencing or exomes or genomes are going to replace tandem mass spec from an economic point of view is sort of ludicrous today. I don't know what it's going to be. I mean, my life in genetics began when there were no southern blots, there was no PCR, there was no cloning, there was no oligonucleotide synthesis. I mean, what's happened just in my lifetime has just been mind-blowing. So I don't know what's going to happen. But to think of it right now, that's just up there as the placeholder. And I'm not going to talk about that. Reduction of false positive results. Sequencing as a second-tier test after a positive biochemical test. And of course, we already do that in cystic fibrosis once you have a positive screening test. And then sequencing as part of clinical follow-up to narrow the differential diagnosis or to really nail the diagnosis and try to bring an end to this diagnostic odyssey that occurs with some of the inborn errors. So this was a study that was one of 4 studies, one here in North Carolina, one at Boston, one started in Kansas and then moved to Rady in San Diego, and then the 4th one was at UCSF, was to explore whole exome sequencing as a complement to tandem mass spectroscopy for newborn screening for biochemical disorders, and to expand the database of pathogenic alleles that we see and to explore genotype-phenotype correlations. Those last 2 are still ongoing research projects, but I'll be able to tell you a little bit more about the first one. So, how was it set up? The California Newborn Screening Program screens about 500,000 live births annually. They get about 300 true positives a year. All true positive children are followed by metabolic centers, and their growth and development, hospitalizations, ER visits are all recorded and sent to the California Department of Public Health. So that's all collected. And this was the way the study was designed. And this was pretty much limited by the funding we got to do the sequencing. If we'd gotten more funding, we would have looked at more because there was more to be seen. But we looked at 1,334 true positives, 394 false positives that were chosen from a selection of over 10,000 false positives, 13 false negatives. Of course, the false negatives, we don't really know how many false negatives there really were. Those are only the ones that were false negative at screen, and then somehow, in the next couple of years, some smart metabolic doc or someone else figured out that child actually has an inborn error and reported it, and may even gone back to the California Department of Public Health and said, hey, how about it? How come you guys missed this? So it turns out there are 13 false negatives, and we did not do any of the true negatives, of which there are 4.44 million total true negatives. This was designed. We extracted DNA from 2 hole punches from the standard Guthrie card type blood spots. Sufficient quality and quantity of DNA was obtained for multiple exomes, multiple libraries to do an exome. They captured and did whole exome sequence and annotated the variants in a set of 44 primary metabolic genes and about another 200 genes that are identified through pathway analysis. The idea being that it may be that there are other genes that are related in biochemical pathways that are not currently known to cause a particular inborn error, but we could discover them this way. And variant analysis was done blinded to phenotype. The entire analysis was done without anyone. Bob Currier held all the cards, and everyone else in the entire project had no idea who was whom. Bob linked the genotype data to the de-identified phenotype data. We looked at the sensitivity and specificity of whole exome sequencing, assessed the sequencing as a method to reduce false positives, and examined variants and correlated to clinical outcomes. So this is sort of the bottom line. The prediction accuracy using exome varied across different metabolic disorders. In green, correctly identified from sequence, and in red were ones where we failed to identify the mutation from sequence. So you can see that for some disorders like MCAD, where there are a couple dominant, well-known variants that are responsible, the pickup rate was very, very good, and was also very good in a number of other disorders. But you can also see that in some, for example, MMA, there were more missed than there were correctly identified. That's probably the one standout of that ilk, or MCD. So, what's the summary of the outcome? The sensitivity for predicting disease from genetic variants had an overall weighted average sensitivity of 88%, sensitivity of 88%, and specificity of 95%. And the sensitivity varied across different inborn errors. So in PKU it was 93%, in 3-MCC 71%, citrullinemia type 1 77%. And I'll get in a minute to why the sensitivity varied, and also what was responsible for the reduced sensitivity. Now, current MBS, according to California's data, is 99.3% sensitive and 99.7% specific. Therefore, DNA sequencing alone cannot currently replace MS screening for these disorders. And that, of course, is not talking about the initial screen. The initial screen is nowhere near that specific. There's a lot of false positives. But this is the program as a whole, including the follow-up, including the extra diagnostic efforts, end up with sensitivities and specificities that are this high. Now, if the analysis of the sequencing did not look at all variants and attempt to figure out, is this a pathogenic variant or not? In other words, trying to do VUS analysis, these variants of uncertain significance. Are they pathogenic or are they not? And really trying to delve into that. If you didn't do that and just looked at already known variants that are known to be involved in the disease or are already known to be benign, the pipeline achieved very high specificity, 99.6%, but the sensitivity dropped down to 58% because you were essentially saying, look, if we don't already know that this child has 2 pathogenic variants, we're just not going to call this a positive. So the specificity obviously takes a big hit. The information storage and analysis was totally secure. There were no results returned to patients. This was done under an IRB protocol that required this. There were no secondary or incidental findings. We did not report, we didn't look for them, and therefore didn't report any back. Now, the California Department of Public Health has very strict rules about what can be done with this data. And one of their primary rules is, if you're doing research that's going to benefit the field of newborn screening, you're going to move it forward and it's going to help the California Department of Public Health do its job, they're okay with that. But they do not want this data shared, even anonymized, even de-identified. So we're in the process of trying to negotiate that with them because there is a wealth of information of pathogenic variants from different ethnic groups. I mean, California is made up of an enormous variety of ethnic groups. And that data, I think, is a rich data source that would benefit everyone in the world. And so we're trying to convince them through the ClinGen project to allow us to do this in a way that does not violate patient confidentiality or privacy. So here's an example of a false negative case. This was a patient who scored positive for glutaric acidemia type 1, MSMS screen. That was the diagnosis during the follow-up, but in our sequencing, we failed to flag GCDH as having pathogenic mutations. These are the 7 variants that were seen. 6 of the 7 have minor allele frequencies in the 1,000 genomes that are higher than 0.25. And so are very, very unlikely to be contributing to pathogenicity just simply by allele frequency alone. One, an intronic insertion A was not present in the 1,000 genomes. There's a software package called Combined Annotation Dependent Depletion, CADD, which tries to use population-based analyses. It's not just evolution or chemical changes of the amino acid. It's actually trying to look at the variant and see, is it underrepresented or overrepresented in populations in a way that would suggest that perhaps it confers some pathogenicity or not. And the genotype for all of these was heterozygous. We only saw one allele. So here's a patient who has glutaric aciduria type 1, and we saw no interesting variants in the GCDH gene in the exome. So why is that? Well, of course, being an exome, the entire gene is not in the reportable range. It basically looked at all the exons, as well as coding and non-coding, as well as sort of intronic regions sort of near the introns. But there could be a deep intronic mutation in that patient that screws up splicing in some way, and you just would miss it. You wouldn't see it. So this is just missed. This is an interesting one. There are 2 patients who were false negative. once again, for this was for IV isovaleric acidemia. The metabolic center diagnosis was IVA, very clear, but the newborn sequencing exomes failed to flag that gene. Just not there, not seen, in either one. Here are the variants that were found. Patient was homozygous for some intronic variants, one of which has a very high allele frequency, the other quite low, and various allele frequency. I mean, none of these would make anyone sit up and take notice. There's nothing about any of these variants. Well, the reason these were missed was when these same spots were subjected to whole genome sequencing, the exon deletions popped right out. And this, I think, is a very important point that I really want to make, is that maybe not this audience, because I'm sure you're all quite sophisticated about this stuff, but I can't tell you how many clinicians I talk to who think DNA sequencing is DNA sequencing. They have no idea that there's a difference between the sensitivity of a panel versus an exome versus a genome. They don't know what you're getting when you're going from a panel to an exome to a genome, what additional information you're getting, and what additional noise you're accumulating. And the entire problem of variants of uncertain significance is just they don't understand. And in particular, the idea that you could have a single exon deleted and an exome just simply misses it cold is well known to happen. And that's what happened here. And the genome picked it up for obvious reasons, is because the genome allowed the split read, the junction to either side of the deletion, to pop right out at you, because you're sequencing along nicely, and then all of a sudden, you're sequencing 4 exons down. Whereas if you're trying to detect exon deletions by copy number, just how many sequences did you get? That's a much more challenging problem. If I get a chance, I'll show you that in a minute. So you can see here, very little coverage in the first 3 exons. Here, this patient was homozygous for a deletion covering the first 3 exons. There's just no sequence here. 5 minutes, great, thank you so much. And then here's low coverage across the IVD gene and exon 12. I actually almost done. So what are the sources of reduced sensitivity? First of all, they're analytic sensitivity. Do we know all the relevant genes? I mean, I think we know most of them. I think we feel sort of comfortable we know most of them. But whenever you do any kind of newborn screening project, you always get surprises. You find phenotypes, either biomarker phenotypes or other sorts of phenotypes, that they're just as surprising. There are hard to do variants, like copy number variants, in relevant genes that are simply missed by sequencing, unless you go to great care to detect them, or you throw a very wide net, such as using a whole genome, which of course contributes a lot of expense at this point. I mean, it's a big decision to make if you're going to use genomes. And then there are intronic variants outside reportable range of exomes that I mentioned before. And clinical sensitivity, of course, also a problem. How do you interpret what's found? It's a coding variant of uncertain significance or an intronic variant of uncertain significance. If you call that variant pathogenic and it's not, you've just messed up your specificity. If you call it a variant of uncertain significance as being benign or just simply to be ignored, but it turns out to be the pathogenic cause, that hits your sensitivity. So how you deal with VUSs in this area is key. So I just wanted to mention this, and I'm almost done. These are 4 diagnoses that were changed by sequencing, and they're kind of interesting, I think. This first one, the screen suggested VELCAD or MCAD, and the diagnosis after follow-up was that the patient was a VELCAD carrier. But the exome sequencing detected 2 mutations in the ETFDH gene, which implicates cloteric aciduria type 2. And those variants were previously observed in 5 adult-onset GA2 patients from 6 different families. So the data that those variants are actually pathogenic are quite high. So it's likely GA2, VELCAD, GA2 overlap in the analyte ranges, which is why this diagnosis was made, but it was wrong. Here's another VELCAD MCAD positive. Diagnosis was VELCAD. Turns out other mutations in the ETFDH implicating disorder again was GA2. These were previously reported in 6 late onset GA2 patients from 5 families. So this is an example of where the sequencing helps clear up a situation where there are multiple different genes which could be responsible. Third one, patient was GA2 positive, but turns out to be had a mutation in the gene responsible for ethylmalonic encephalopathy, ETHE1. And then finally, a patient was said to have isovaleric aciduria, and that turns out actually to be a patient who has 2-MBCD, methylbutyryl-CoA dehydrogenase deficiency. Once again, so it all has to do with the fact that there's overlap in the analytes that resulted in these patients being given a incorrect diagnosis. And with this, I'm going to end. This is the group that has worked on the MBC at UCSF. I want to point out in particular Ashish Adhikari, who works with Steve Brenner at Berkeley, and this is the group that's really done the bioinformatics. This is the group in the administrative section, Jennifer Pucks, the PI, Pui Kwok, and Barbara Koenig in our bioethics group, and these are the drones down here. Just kidding. These are wonderful people. Renata Gallagher is a tremendous contributor, and Joseph Shea in the metabolic side. Neil Risch, who's just a superb human geneticist. Sean Mooney, who's been working on mutation detection. So with that, I'm going to stop and take any questions.
Just quickly, so in the cases where you missed the review, they were not even identified as heterozygotes?
Right.
They were not even identified as heterozygotes?
That's right.
Wow.
They found no variant that was the slightest bit sort of curious.
Wow.
The allele frequencies were too high, and there was nothing about them that suggested they'd screw up splicing.
Wow. And so, and then the 5 cases you just showed.
Yeah.
So you went through the same system that Mayo used, that Ronaldo developed, and they didn't pick those up.
Yeah.
And showed no suspicion of it.
Yeah.
And even retrospectively looking at it, you saw no interpretative mistake.
You know, I don't know if we've done that or not. I need to, because that would be something we need to go back and say, hey, you know, we actually have confirmed pathogenic mutations in these other genes. Go take another look. We should do that with Piero.
Yeah, because I mean, with Piero, he should have a comment on that.
Yes.
I'm sure he'd be interested.
Yeah.
Thank you for a very nice lecture. I have always been curious the advantage of looking at through the sequencing, if we could pick up diseases which are not on the MSMS panel, like glycogen storage diseases or some of the urea cycle, which have the intervention, and yet due to the not, you know, did you look at any of those disorders like PDE?
Yeah. So we had one patient who had a newborn screen that looked like possible OTC deficiency. It was a female. And then she was followed for 5 years. Nothing showed up. She never had any hyperammonemia or any problem at all. So she was finally told, that's false positive. She's a carrier for OTC deficiency. So that was picked up. There's no question that we can pick up all kinds of stuff by sequencing. The reason that we focused on this was we have the power of a biomarker and 5 years worth of follow-up. So we actually have what you desperately need when you're doing a sequencing test, and that is a gold standard. You in essence, know the truth about that patient.
Thanks very much for a great talk.